if you look at the genetic material, amount of DNA, remember when we are talking about cell division, we have to talk about what is the number of chromosomes, and we have to talk about what is the amount of DNA in a given cell, right? Now here the number of chromosomes should be measured by the number of centromeres, right? Now in this early cell, before it enter into meiosis 1, right, this early cell, Spermatogonia, the number of chromosomes is 46, and each chromosome has single structured. Let me explain this. Let's suppose this is chromosome from my father, and here is chromosome from my mother. Let's suppose these chromosomes are homologous to each other. When I say these chromosomes are homologous to each other, it means that both of them are having alleles of the same genetic material. For example, if it is chromosome number 3 from father, paternal, then it must be chromosome number 3 from maternal side. And if here is a gene in the DNA, of course, what is there? There must be double-stranded molecule of DNA. Every chromosome, look, this is double-stranded molecule of DNA. And if these chromosomes are homologous to each other, right? Homologous to each other mean chromosome 1 of mother is homologous to the chromosome 1 of the father. Chromosome number 10 coming from mother should be homologous to the chromosome number 10 of the father. Is that right? So corresponding chromosomes from mother and father are homologous to each other. X and Y are not homologous because they are not having the same genetic material, right? Now listen. Now these are one pair of homologous chromosomes. Is that right? Now in this case, both chromosomes are having the similar DNA, and in the DNA there are genes, right? What are genes? Genes are sequence of nucleotides which make a specific functional RNA. Now let's suppose if there is a gene here, right, and this gene is about the color of the eye. Now this gene which is present over here, it determines the color of the eye, and this gene is coming from the mother. Then on the same homologous chromosome which is coming from the father, at the same locus, at the same point, there should be gene which also determines the color of the eye. And here is suppose another gene which determines the size of the nose from maternal side. Then on this homologous chromosome coming from paternal side, on the same locus, right, from paternal side, there should be gene, and this gene should be also determining size of the nose. For example, here there is a gene which determines structure and function of a specific enzyme, right? Then on the corresponding locus on the other homologous chromosome, there should be a gene which should be responsible for the structure and function of the same enzyme, right? Now these genes, look, these genes which are going to do similar function, and they are present on a homologous chromosome at the same loci, right? These genes are in specific relationship to each other. As I told you, this gene is making color of the eye, maternal side gene, maternal gene for color of eye. This is paternal gene for color of eye. Both of these genes are alleles of each other. What are they? They are alleles of each other. And what is this? Gene for the suppose size of the nose or shape of the nose from father side. This is the gene for the shape and size of nose from mother side. So these two genes are also alleles of each other. Then if this was a gene from paternal side for a specific enzyme, and here on the same locus on the homologous chromosome coming from the mother side also has the same similar gene for the same enzyme, then these genes are also alleles of each other. So what we can say, when two chromosomes are coming from mother and father, is that right? And they are having similar genetic material, such chromosomes are homologous of each other. And on the homologous chromosome, the genes which are having the same loci in their position and doing the similar function, right, or dictating the similar genetic information, these genes are what they called alleles. Is that right? Any question up to this? No problem. Now, you must remember also one more thing. These are the chromosome, let us suppose chromosome number 3 paternal. This is called single structured chromosome, right? This is also maternal chromosome, but this is also single structured. When chromosomes replicate their DNA, chromosomes become duplicated. Let's suppose this chromosome, right, it replicates its genetic material. When the DNA replication will occur, what will happen to this chromosome? After DNA replication, this chromosome will become duplicated. Is that right? In the same way, if DNA has been replicated in this maternal chromosome, this maternal chromosome will also become, yes, duplicated. Now, here it is very important, in duplicated chromosomes, right, of course, DNA material is double. Is that right? Because DNA has been, yes, replicated. Replication of DNA lead to duplication of chromosomal structure. Or we can use another term. Now listen, this chromosome which does not have replication of DNA, this is called, it is one chromosome, one copy of chromosome number 3, and it is single structured. It is also one chromosome but double structured. Why? Because chromosome number is basically counted on the base of centromere. Is that right? Centromere is the point where the chromosome is constructed. Is that right? Now, so this is single structured chromosome, this is double structured chromosome. Single structured chromosome is unreplicated DNA, double structured chromosome has replicated DNA. Is that right? Single structured chromosome is also called chromosome with one chromatid, and this is also called one chromosome with two chromatid, right? Or we call this unduplicated chromosome, and this is called duplicated chromosome. Let me recap. When one chromosome has one centromere and one short arm and one long arm, what we call that? This is unduplicated chromosome, or it is chromosome without replication, or it is single structured chromosome, or we can call it chromosome with one chromatid. Is that right? But if this situation is there, the DNA of the chromosome has been replicated, it is still one chromosome. So what we'll call this? One chromosome with replicated DNA, or we can call it one chromosome with duplicated structure, or we call it one chromosome with double structure, or we call it one chromosome with two chromatids. So I hope you will remember these terms. What terms I have made clear in your mind? What are the homologous chromosomes and what are alleles on each other? Is that right? Homologous chromosomes have homologous alleles, and then I talked about what are single structured and double structured chromosome. In my lecture, I will continue such chromosomal structure as single structured chromosome, and such chromosomal structure I will talk about double structured chromosome. Any question here? No. Now let's move forward. Let us suppose that in my body spermatogenesis is going on, and if spermatogenesis is going on, it means my germ cell must be undergoing the process of meiotic division. Which division? Meiotic division, which is a specialized type of division which occurs especially in formation of germ cells and gametogenesis. Is that right? Now, let's suppose this is in my testes. This is the primary cell. Of course, my testes will not have oogonia, right? Hopefully. Now we come to Spermatogonia B. Now, Spermatogonia B in my body, it has 23. Let me write here. It has 23 maternal chromosomes. 23 maternal chromosomes. Plus, it has how many chromosomes from paternal side? Also, yes, 23 paternal chromosome. Now, when there is a full set of 23 chromosome, we call it 1n, one small n. So we will use a term here that in this cell, so Spermatogonia, there is number of chromosome, one small n from which side? Paternal side, one small n from maternal side, right? One small n is equal to 23 number of chromosomes, one set of 23 chromosomes. Now, if you put them together, right, how many n's are there? It is 2 small n. And when there are 2 small n, it means it is double of 23. So we can also say that this 2 small n is 46 chromosomes. Or when number of chromosomes is 2 small n, or 46, we say it is diploid number of chromosome. What we call it? Yes, diploid number of chromosome. I'm talking about this cell which is about to enter into meiosis 1. Now, another thing, this was about the number of chromosome. Let's talk about the genetic amount of DNA. Is that right? Now, amount of DNA, this is single structured or double structured? Single structured chromosome. And what is this? This is also single structured. Now come back here. In the single structured chromosomes, if there are 23 chromosomes which are single structured, 23 chromosomes which are single structured, we call it the amount of DNA is 1 capital N. Capital N is notation used for the amount of DNA in a given cell. Small n is the notation which is used to the number of chromosomes. Now, if we talk about there are 23 paternal chromosomes, single structured, what we will say? That there are 1n paternal chromosome and 1n mean 23 chromosomes. And I say 1 capital N also, right? This is not only 1 small n, it is 1 small n and it is 1 capital N. Now, when I say it is 1 small n situation, it means there are 23 paternal chromosomes. And if I say 1 capital N, it means it is single, all 23 chromosomes are single structured. What is this? Single chromatid or single structured or single chromatid chromosomes. Is that right? Now, in the same way, these 2 chromosomes I have drawn here, right? Again, it's maternal side. How many chromosomes from maternal side? There are again 1n from maternal side, so 23 chromosomes set from maternal side. And each chromosome is double structured, single structured here. Single structured, so it will be 1n, single structured. Is that right? So in this case, what we will say? That 1 small n plus 1 small n. So what is the number of chromosomes? 2 small n. Or we can say, what is the number of chromosomes total? 46. So this cell has 46 total number of chromosome and diploid cell. But look at the DNA of it. It has 23 single structured from paternal side. It has 23 single structured chromosome from maternal side. So 1n from maternal side, this is amount of DNA. So amount of DNA from mother is 1n. Amount of DNA from father is 1n. So how many capital N amount of DNA? 2n. Is that right? Any question up to this? No. Now we come to this situation. Actually what happened, before this cell is entering into meiosis 1, before this cell enter into meiosis 1, it is going to replicate its DNA. And all chromosome, paternal chromosome as well as all maternal chromosomes, they will replicate DNA. When all the DNA is replicated, all the chromosomes will become double structured chromosome. What they will become? Double structured chromosome. It means when these single chromosomes will become double structured chromosome here, so these structure will convert into this type. So it means every chromosome is a double structured. Now this cell with double structured chromosomes, right? This is called, yes. What is this called? It is called primary gamete. What is it? What it is called? Primary gamete. Of course, this primary gamete in female it will be primary oocyte, and of course I'm not female, so it will be primary spermatocyte. Is that right? Now, in the primary spermatocyte, which is derived from Spermatogonia B, or in the female, primary oocyte, which are derived from oogonia, right? They are having double structured chromosome because they have already replicated their DNA. And if they have already replicated their DNA, now very carefully we'll talk about the structure, right? How many paternal chromosomes are there? Now we have 23 paternal chromosomes but double structured. Here we are 23 paternal chromosomes with single structured. So what we'll say that paternal chromosomes are 23. How many paternal chromosomes? Paternal chromosomes are 23, and how many n? 1n. one small n. Is that right? And plus on maternal side, yes, maternal are how many? Look, at this cell there were 23 single structured, and here there are 23 double structured. So we will again write 23 but double structured maternal chromosomes. Is that right? And how many small n it is there? It is still one small n, because even though they are double structured, but actually the number of chromosomes depends on number of centromeres. Is that right? So how much it will make? Both of them together. Total chromosome will become 2n small. Or we can say total number of chromosome will be, yes? No, it will be still diploid. This cell in the beginning will be still diploid. But there is a very important thing. Amount of DNA has been doubled here, right? Because every chromosome from here to here or from here to here has become double structured. Now, if we are talking about the amount of DNA, now here the amount of DNA was 2n. 1n was contributed by father and paternal chromosomes, and 1n was contributed by the maternal chromosome. Is that right? And in the same way here now 1n has become 2n. So 2n in paternal set of 23 chromosomes. So we can say that paternal, this is actually to capital N, amount of DNA. Here it is how much? Again 2n, because it is also double structured. So total n will be how much? 4n. So we can say that primary gametes mean in males primary spermatocytes and in females primary oocyte, right? All the chromosomes from maternal and paternal side are double structured chromosome, but number of chromosomes still remain diploid. Is that right? But amount of DNA has become double. Is that right? Any question here?